other questions? Okay. Thank you. Okay, so the last talk is by Patrick Underhill from Rensselaer Polytechnic Institute, and he's going to tell us about velocity and stress correlations in suspensions of swimming microorganisms, theory and simulation.
I'll try and figure out how to get it so that it actually shows up the whole thing and not just the right side of it. Yeah. So,
Put that in your pocket.
So I'm in the chemical engineering department at RPI, and I guess in most of the research I do, hydrodynamics plays a pretty key role. So I'll be talking about some of the hydrodynamic effects in the correlations in swimming microorganisms, and we've heard a number of talks about that, so I'll skip quite a bit of the motivation. But in addition to sort of what leads to the collective behavior, I'm also interested a lot in the biological sort of relevance and impact of these. So I'll first show you one experiment and describe it, and then I won't talk about it anymore because it's too hard for me to do right now. But it's sort of the final goal I sort of have in mind. So this is a real-time movie of E. coli swarming on an agar plate. So we're looking down on top of the plate of agar. So essentially what happens is, right, you take your suspension of E. coli and you put it down on this plate of agar. And first, sort of a biological thing happens, which is they sense something about their environment. We don't really know yet what it is. And when they grow, instead of dividing, they don't divide anymore. They just keep getting bigger. And they have more and more flagella. And then once they are in that state, which is called a swarming state, you see these patterns. And as you've seen probably as this loops, the front of this group of E. coli is moving. So that allows them to very rapidly fill this entire plate of agar. And so perhaps this has some biological relevance in terms of sort of invasion of different regimes as sort of the bacteria are trying to, say, invade your body or something like that.
Everyone gets bigger this time?
So,
Are they eating?
So they are eating and they are dividing, but right, this is in real time, so you're not this is not driven by sort of their growing, right, because you're looking at it over a pretty short amount of time. But before this happens, they do have to grow and get larger and get many more flagella. So these are just the body, right, that you see of the E. coli, and it's pretty densely packed. And the flagella are sort of all intermingled, and it's kind of amazing that they can actually move with their flagella being so sort of tangled up. So that's the last I'll say about that, and move to a much simpler geometry. And so what I'll be talking about is correlations in these suspensions of these swimming bacteria. And so the two types of correlations I'll talk about are velocity correlations and stress correlations. Hopefully I'll get to sort of the end and say a couple slides about the stress correlations. So the velocity correlations, there are two types of velocity correlations. So suppose I have these two sort of organisms, right, which have label 1 and 2. So there's some direction that it's trying to swim in, and then there's some velocity that it actually goes in. And those are not necessarily aligned. And then there's another sort of swimmer with some orientation and some velocity that it goes in. So I'll be talking about the orientational correlations of how correlated N1 is with N2. Also, you can look at V1 and how correlated it is to V2. But this is also in a fluid. For this, we'll be talking about a Newtonian fluid. And so there's some fluid elements, say with some fluid velocity, and over here the fluid velocity might be like that. And so you can look at how correlated these fluid elements are as a function of their separation. You can also, right, as we've seen before, this active sort of motion of these organisms induces a stress like this at this position. And there's another stress here in this position. And so you can look at the stress as a function of time, or space and time. And then look at spatial correlations of the stress, and also look at time correlations of the stress. And so sort of one reason we're doing this is to try and quantify some of these sort of collective behaviors, like these swirling patterns. Rather than looking at them and seeing they're similar, we wanted to try and quantify them by using these correlations. So the velocity correlations, we're going to try and use to quantify this collective behavior. But these velocity correlations can also sort of be related in some level to maybe the short time 2-point microrheology. So suppose you are doing 2-point microrheology, which essentially you track the motion of 2 probes that you put in. At short time, this probe might be going in that direction and that probe in that direction if they're being advected by the fluid. So we're neglecting sort of the Brownian motion right now of those probes. And so at short time, these velocity correlations will tell you something about the 2-point microrheology. Recently, there have been some theories to look at sort of the long time behavior or the spectrum of the 2-point microrheology and how that is related to these stress correlations, which is why, in particular, we are interested in that. So this is sort of the simplest model that we use to do our simulations and our theory. And so I might just say a few words about this since it's a little different, but similar to what we've seen before. So the simplest model that we have is to take this, say, organism, for example, E. coli or B. subtilis, and represent it by 2 beads connected by a rod. So this is very different from, say, what Aparna was talking about, because these 2 beads are the same size. And so if you looked at sort of Aparna's swimmer, that wouldn't swim, whereas this does swim, which might be kind of odd, because it's totally symmetrical. And so I want to sort of describe how that works pretty briefly. So essentially we treat the body of this organism as a sphere. This is sort of a real sphere. So with an E. coli, the body of the organism is, say, moving to the right. And so if this bead on the right is moving to the right, it has to push fluid out of the way. Right? A sphere moving through a fluid causes a disturbance. So that's what this blue line represents, is the sort of disturbance in the fluid caused by this organism. Now, this back sphere, I draw it as a sphere, but it's not really a sphere in some sense. It acts like something which is not a sphere. So if you take sort of flagella, right, it's a helical filament which rotates, and that's what causes the motion. So suppose you just had a helical filament without the body of the organism, right? And you somehow apply a torque to it to rotate it. So it's going to translate through the fluid. So now, suppose you tag or sort of attach to the front something that has to push through the fluid, right? It can't go as fast as it would want to. So it's going slower. So the disturbance it makes is as if it's going backwards. So it pushes the fluid backwards, even though it's going forwards. Right? So that's kind of an odd, a sphere wouldn't do that, right? A sphere makes a disturbance in the direction it goes. But a helix can make a disturbance backwards even if it's going forward. So that's why the swimmer can swim. The flagella just pushes backwards, the body pushes forwards, and it sucks fluid in from the side. So hopefully that didn't just confuse you more. So then we basically, right, similar to what we saw earlier, if you have another organism over to the side, it will be ejected sort of towards or attracted to this other organism. And so we essentially put a lot of these organisms into some domain and follow their motion. And I'll also mention sort of the effective volume fraction is the number density n times the volume of a sphere with length L over 2. So these two B's are separated by a length L. And so this is a movie showing sort of one of the simulations. I essentially, in these results, take a 3D periodic box, and I randomly place these organisms in there, and then I follow their motion in time. And I do that by neglecting inertia, so I write down a force balance. And then there's a hydrodynamic force, which is just a drag. It's minus a drag coefficient times the velocity of particle 1 minus the velocity of the fluid at the position of particle 1. And this fluid velocity is basically generated by all of the other organisms. Other than the swimming, it's a perfectly stagnant fluid. You're only getting fluid motion because of the swimming of the organisms. And so this, the way we formulated it, as sort of mentioned, sort of as a comment to Aparna's, the way we formulate this, we could do this as a pairwise additive calculation, but that still captures the multi-body hydrodynamic interactions, which is a slightly subtle point. And the other thing to mention here is that sort of in comparison to these local interactions that you might have, these are very global long-range interactions. And so we have to include every possible pair. And we do that in a very efficient way, which I won't get into. So here are the things I leave out, so I won't go into that. You can ask me later. It's a lot of things. So I'll skip that and just go into this. So we quantify the fluid structure by doing a fluid velocity or tracer velocity correlation function. So it's the fluid at some position dotted into the fluid velocity at another position r away. And we calculate that correlation as a function of the separation of the two fluid elements. So I have three different cases on here. The blue curve is if these organisms are not interacting at all, but they're still disturbing the fluid. So they just swim straight forever without sort of seeing one another. And they still have a fluid-fluid correlation, which is essentially most of it is captured by this black line, if you believe me for a second. And so that's another, if you're asleep, pay attention for this 1 second. So in contrast to some of these other correlations we see, you might expect to see like an exponentially decaying correlation with some correlation length. That's not what you see in hydrodynamics. Even if they are not interacting, there's no collective behavior, the fluid is still correlated with 1 over r correlations. Right. So it's a very subtle thing to measure the collective behavior. So if you have, as Aparna was mentioning, there are these pusher-type organisms and puller-type organisms. The puller-type organisms are interacting with one another, but that is not captured in this fluid-fluid correlation function. It's essentially the same as if they weren't interacting. But pushers do have some sort of larger correlation in the fluid. And that also increases with system size. So we have some finite size effect as we make the system bigger and bigger. And you can capture that with this theory. So asymptotically, you get this theory, which is 1 over r. That's the independent contribution. And then there's this finite size effect, which is the log order phi squared it's a pairwise term, but it's logarithmic in the size of the system L over the separation. And using these two sort of fitting parameters, we get to see that sort of system size dependence. And I'll skip the stress correlations, other than to say that in our theory, we see a Lorentzian as a function of frequency. So a high frequency goes like 1 over omega squared, whereas experimentally, it goes like omega to the minus 1/2, and I don't know why that is. So I'll leave you with that and try to take any questions.
Thank you. Any other questions?
Or did I totally confuse you?
Over there?
You have finite size effects. If you change the size of your system, it changes the properties.
Yes.
What did you try doing periodic boundary conditions to simulate like that?
So, sorry, I had to go through that so fast. So, this is periodic boundary conditions. So, I'll just put up the simulation. So it is periodic boundary conditions, where L is the size of the box. And so what we do is we say change the size of the box, but keeping the volume fraction the same. Right? So in principle, you'd want if
Size you meant
to yes. So it seems like there's essentially infinitely large correlations in the system, which get cut off by this imposed periodicity. And as you sort of relax that by making your box bigger and bigger, you're allowing these correlations to get bigger and bigger and bigger. And those grow sort of logarithmically.
You said you wanted to include run and tumble dynamics.
Yep.
In a swarming system, do they still run and tumble, or do they just go straight and go in big swirls?
It's not really clear, I think. So I think in some of these systems we could it might not be that important. So, right, if you have some organism which it gets reoriented because it is either moved by the fluid or hits another one, sort of before it has time to tumble, then the sort of run and tumble might not really play much of a role. And in sort of that experimental sort of the swarmy, they're sort of so concentrated and they're changing their direction so quickly that I think sort of the run and tumble might not be that important. So then it might sort of raise the question of can they sort of chemotaxis, right? Can they? If the run and tumble doesn't affect their motion very much, then can they still move to, say, higher concentration or something?
Okay, so I think it's time now for the discussion period. We're going to shorten the discussion.